I'll just present the slides and go through what's on this list. I should have 6. I'm missing one. Oh, no, I'm good. Okay, so I've got them all here. It may help to turn the lights off, just at least in the front. So we can see the image better on the scope. So what I'm doing is I have the scope here, and what you're seeing is what I see there. And if you recall, microscopes have, well, there's 4 objective lenses. We're going to use 3, low, medium, high. This is a low view. I'm going in order. I know it's kind of hard to read in the dark. But the thyroid parathyroid, I think that's the first I have listed. Let me do a couple of other logistical setups here. I'm going to get my mouse over by the microscope. Got to be able to click the camera icon if I want to take pictures for you, so bear with me. This is so sophisticated, not good. Oh, that's good enough. We'll go like that. All right. Now let me pull up a chair. Now, I haven't taught parathyroid yet. Where's my pointer? Because if I did, you kind of know what you're looking at here. You guys see my pointer? Because I'm totally discombobulated. I'll just point with this ruler here. Oh, the ruler's gone. I'll just point with my fingers. Okay, wherever you see, this is a low magnification, and the thyroid and the parathyroid are together on the same slide. What looks like solid, dense, packed cells, dark purple or light purple, here and here is parathyroid, and everything else is thyroid. So this is a low magnification. Let me I already took a picture of it. Let me turn the lights on so I can kind of draw something. Maybe that'll allow me to find my dang pointer. Going crazy. I think I have another one somewhere. Okay. Oh, here it is. Jeez, there's my paper. When we go under a high magnification, what's unusual about the thyroid is you secrete the hormone in an inactive form, but you have to keep it in the gland, but outside the cell. Most cells don't do that. They can just make their hormone and secrete it, it's good to go. But the thyroid requires iodine, and iodine can't be made by cells. It can only make proteins. So you have to extract it from your diet, from your blood. And so there's a process where the follicle cells will secrete an inactive form of thyroid hormone and keep it in the middle. And when you cut a follicle open, what you kind of see is a bunch of simple cuboidal cells we call them follicle cells. Not to be confused with the follicle cells in the ovary. but they secrete their inactive hormone in the middle. I'll color it red. It's red gelatinous mass. We call that red mass colloid. Colloid is, think of it as inactive thyroid hormone. Okay, in histology we call it colloid. Okay, so that's kind of the general structure. So that's why on the PowerPoint, on the microscope slides, not a PowerPoint, you see all of these masses in the middle. You can even make them out at low magnification. Now, when we go to high magnification, we're going to see these little clumps of cells outside the follicle I drew them as blue cells. They're called C cells, or we can also call them parafollicular cells. They secrete the hormone calcitonin. I'll lecture on that on Wednesday. But I didn't lecture on this, so I at least wanted to give you some background. The follicle cells will secrete the thyroid hormone. They just have to extract the iodine from the blood first. And I'll show you the physiology of that later. But for now, let's just see if we can identify these things in the thyroid under a high magnification. Now, before I leave this picture, I said the parathyroid, the dark pieces, and the names of the cells for the parathyroid gland, well, the chief cell is what they call it the chief cell. It's the cell you see the most. So for what I drew here, this is all thyroid. That's all thyroid. Okay, so, but for parathyroid, I guess know the name chief cell or principal cell. I'll draw a little arrow here. Chief cell. The chief cell secretes parathyroid hormone. That should be in your notes from yesterday. Or not yesterday, but on Monday from the first class. So let's go to high mag. I'm going to turn the lights off. This is 40x. I call this the low mag. This is 100x. I guess we'll call this medium magnification. Okay, but you really got to go to 400x to see cells. So I'll just go to 400. Turn up the light. It's pretty good. Okay, I'm going to take a picture of that. So we can point out what we're seeing here. Inactive thyroid hormone in the middle. Any of these, that's colloid. Okay. If you see cells surrounding the colloid, call those follicle cells. If you see cells, it looks like they're outside the follicle, call those the C cells. Okay? Or like here, like that one. It's a larger cell. The C cell means clear cytoplasm. So on some of these cells, they kind of look like they're kind of faded a little bit. If I point to any cell outside the follicle, just go with parafollicular cell or C cell. Okay, moving on. Any questions on this slide? The next slide is, we did thyroid parathyroid. Let's do hypothesis. Let me get my little slide cleaner here. This one's a little dusty. Okay, let me pull back a little bit. Whoa. What is that splotchiness? Yeah, that's, geez.
Oh, it looks like condensation. It'll kind of go away.
Let's see here. Let's get that fixed here. Yeah, maybe we got something on the lens there. Good. Let's go. Okay, I'm going to pull back to this lowest magnification. This is about a 20x. Okay, see that little light strip of light staining tissue? That's infundibulum. This is continuous with the posterior lobe. So if it were cut right down the middle, you should be able to follow that all the way to posterior lobe. So I'm going to follow it down. Keep a little bit of it in the frame of view. I'll take a picture of this. This is a low magnification. Infundibulum, which is continuous with the posterior pituitary, pars intermedia, anterior pituitary. That's it. Okay, I'm going to move on. Any questions on that one? About to come off the stage here.
Posterior is typically lighter, you said?
Yeah. Mm-hmm. That is correct. Posterior is the lightest stain, and what's the darkest? Anterior. And pars intermedia is an intermediate stain. Okay, moving on. Is there anything else? No, that's everything. I'm looking at the study guide. That's everything. I'm going to proceed. Pancreas. The pancreas, I didn't teach that yet, but I'm concerned with the blood sugar regulation, and those hormones come from these little clusters of cells, these little islands of cells, or islets. So that's why you got to be able to identify pancreatic islets on pancreas. Here's low mag. Put that in the magnification. I'm going to go to a medium magnification. This is a 100x. So most of the pancreas is for exocrine function, for digestion. But the endocrine function, the cells are very clumped together in little islands to regulate blood sugar. They'll secrete the insulin and the glucagon. They're called pancreatic islets. And I'm doing it for you today, but Friday's lab is you're going to show me stuff on these same slides. And see these little cluster of cells there, or there, or there, or there? Those are the islets, okay? Pancreatic islets. This is something else, probably pancreatic duct. That's probably part of pancreatic duct. That's probably a blood vessel. That's cool, because let's remember, hormones need access to a bloodstream. So let's see if I can get that into a high mag. Pancreatic islet. They secrete insulin and glucagon. If you have type 1 diabetes, you destroy those islets. Not very many of them. We get in the center of the field of vision. And we're going to go to a 400x. That's the highest mag I will allow students to use. That's pretty good. Take a picture of that. So the key for you to understand is what's the islet, what's the blood vessel. I recognize the thin vessel wall there, the small blood vessel. Those are the red blood cells. This is what we're identifying. See the little nuclei? There, in this little light staining clump of cells, that's the pancreatic islet. Okay? They secrete insulin, glucagon, and we'll move on. It's pretty straightforward. Okay, adrenal gland. All right, see if you can identify the border between cortex medulla. And see if you think you got it, and then I'm going to point to it. That's right here. Is that where you're looking? So above it is what? Above my pointer is what? Cortex. And below it is medulla. This is the short-term stress response, long-term stress response. Okay, I may have you identify this down here as adrenal medulla. You got the 3 zones. If I point to the outermost zone, is this a zone?
No.
That's the capsule. That's not a zone. Okay. Zona glomerulosa, zona fasciculata, zona reticularis. Okay. See the redness in between the cells? That's like blood vessels probably. But anyways, it's the salt sugar sexting. Okay? So study that. Adrenal gland. Done. Moving on. Okay, ovary.
Is the capsule part of the cortex or is it not even?
No, it's not part of the cortex. It's just capsule. Yeah. So this is a low magnification. This is a cat ovary. It's good for teaching because it's got like, well, when cats, you know, they have litters. They have like a lot of kittens in one birth. Humans have one. That's it. But when cats, they have a litter. So there's a lot of different varying stages of development. So we can see them all in one shot. See all these little circles in the edge? That's our primordials. Okay? You can even notice that under a low mag. You see the one right next to it's kind of bigger? What's that one going to be? Primary. And the secondaries are easy to see. You got one graphene with 2 eggs in it there. But there's a nice graphene right there. Let me see if I can get this into a higher mag. Let's go from low. This is a 40x. This is 100x. Go to the edge. So this is 100x. We can see our primordials, our primary. That's a larger primary. That's a graphene. I don't see a really good secondary in this field division, so I'll go one more. Look around, look around. Oh, these are good secondary. Okay, so, I see 4 secondary right here. This thing right there, boom, 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 boom. Those are all secondary. Secondary, okay. Now, if you even like zoom in right there, maybe we can see the theca granulosa cells. go white balance. That did not help. That's okay. All right, so there's the OO side of a Graphian. But I took this picture so you can identify these cells, granulosa cells from here to there. That's theca. These secrete the androstenedione, transferred to here, converted to estrone. Okay, under the influence of FSH and aromatase. But anyways, I'll just have you identify things. Theca, granulosa cell, that's it. Okay, moving on. Any questions on ovary? You got one to go. So you see a bunch of cut tubes, and you see like kind of stuff in between the tubes, so to speak. I mean, stuff isn't a scientific word, but seminiferous tubules, all cut. Okay, but all the things in the middle there, in the interstitium, those are the cells that are like theca. They're going to secrete the testosterone, the androgens. We can even see blood vessel there. This slide stains the red blood cells black. So when they're in the lumen and they're black, I know it's a red blood cells in a blood vessel. So I'll put this under high mag. All right, so I just say, this is seminiferous tubule. What if I said identify cells? Interstitial cells, that's it. It's pretty easy. Histology quizzes aren't that hard, actually, if you take some time with the slides. All right. Got them all there. I can put these images on the PowerPoint, and I'll upload it on Canvas. I guess one thing I'll say, because I always get asked, I'm not going to label the slides for you, but I will use the same slides for the quiz on Friday. You have any questions about the quiz for Friday?
If you want us to name a specific place, will there be an arrow pointing to it?
Yeah, I'll point to it.
Okay.
Yeah, it'll be a PowerPoint format. If it's not obvious, just ask me during the quiz. What do you point at? Yeah.
And it'll be fill in the blank.
Fill in the blank. So spelling counts. Yeah.
Are you gonna post these today?
Yeah, this afternoon. Yeah.
Do you just label the part, or do you have to say like that's the blood vessel of the testes?
Oh, well, like for example, you can just say interstitial cells, and that'll be it. You don't have to put of testes. Yeah. If there's no more questions, class dismissed. I'll see you Friday.